Good morning, everyone. This is Dr. A.V. Siva Kumar Reddy, working as Assistant Professor in Department of Physiology, Narayana Medical College and Hospital in Andhra Pradesh. Today, I would like to talk on reason for failures in medical innovations. Before I take you to this discussion, I would like to express my sincere gratitude to the respective management authorities for giving me this opportunity to express my view on the reason for failures in medical innovations. I start this discussion. Let me introduce myself. I am Dr. A.V. Shivakumar, completed my Master of Sciences in Medical Physiology from Narayana Medical College. Later, I completed Doctor of Philosophy in Medical Physiology from Sri Ramachandra Medical College, Chennai, and I am working as an assistant professor in the Department of Physiology, Narayana Medical College, Andhra Pradesh. I have acquired knowledge in evaluation of non-invasive screening tools for their validity and reliability and other neurophysiological as well as cardiovascular variables like heart rate variability, pulse wave velocity, dynamic pupillometry, the simple reaction times, and critical flicker fusion frequency. I have published more than 35 research papers in various index journals, and I have also received 10 national awards for the contribution of scientific knowledge in the medicine. And I am also honored as an editorial board member as well as reviewer in various national and international journals. The James Bond movies where they used to project various kind of fascinating innovative gadgets of the uh, James Bond, where that has come to the reality. So all the directors of the James Bond series have this quotation, that is, today's science fiction is tomorrow's science fact. That is, when you see in the olden James Bond movies where they show the cameras, recording cameras, and the voice recording instruments, the speech recognition devices, the automated driving cars, whatever you see in that particular movies, it has come to the reality because of the advancement in the technological field, which has even extended into the field of medicine. Reality, all the medical innovations or whatever the innovations they have come out may not be succeeded in terms of execution, in terms of point care use to the people as well as into the society. Because the plan or the ideas which may have the technical issues when it comes to the execution, or it may be too much advanced to this generation or to understand that particular idea by the expertise. So that is why most of the medical innovations or the real-time innovations in different fields has been failed. So it doesn't mean they have failed, rather it is a part of learning. So to change the practice of medicine, the culture of an organization must not only tolerate but also welcome failure as a part of the learning process. So I would like to discuss this topic under these headings. So first of all, what is creativity? How it is different from innovation? What are the stages of innovation in healthcare system? How to assess failure of medical innovations? The types of failure in medical innovations, and I'll give you one example where you can able to understand what are the possible causes or the reasons for failures in medical innovations. So if you look at the healthcare system is transformed in the past few decades in terms of developing screening tools, the diagnostic instruments, the preventing instruments, as well as therapeutic regimens due to substantial advancement in technological innovations. So now, even the doctors are well prepared to become entrepreneurs because they are the professional problem solvers where they understand the requirement of the patient and they also understand the current trends in the healthcare system and how to overcome the day-to-day issues. But the problem is all innovative ideas do not succeed due to failure in any step of execution as well as results. I mean, it results in refinement. So here, whatever the ideas are getting by the doctors, it requires substantial support from the collaborating departments where it has to be properly evaluated, properly executed to produce the reliable results. If they don't get it, obviously it results in the failure of the innovation. See, before I talk about the failures in the medical innovations, first of all, you need to understand if you get an idea to innovate one medical device or one medical instrument, you should know what are the rights you have for such creative innovation to be implemented in the field of medicine and how responsible you are for that innovation, why such innovation is required in the healthcare system, which means what is the rationality and the need of population in the healthcare system. See, here I would like to project the difference between what is creativity and innovation. See, the creativity is the generation of new ideas, whereas innovation is converting that creative ideas into desired outputs. For example, if you take in the teaching abilities, initially we used to draw the diagrams on the board and we used to explain everything which was so messy to the students as well as to the teacher. But now we got the PowerPoint. We used to change initially the slides using the keyboard, but later on we got the slide changer, which will be controlled using infrared light rays. So slowly there is a transition of the virtual teaching as well as audio visual aids, which are substantially improvised in order to facilitate the student for their perception. So the idea, what we get, has multiple stages. So initially, when you get an idea, so-called the creative idea, we'll think of how much possibility it has to make it as the innovation. So we'll try to share to the maximum people where they will express their own suggestions or the comments with which we may fall into stage two, that is doubts, whether it is possible or whether it is feasible, whether it is executable, then we enter into a stage called agony. So we will not be in a position to express our creative idea because we thought that it will be criticized, it will be made fun of us. So we are not in a position to express this. But slowly, as we correct our idea, as we improvise our idea, at one point, instantly we can overcome all these anticipated problems. Finally, we have an answer for every critical comment of the expertise, so it is ready to be executed. Then we'll be in a position to say Eureka. So in order to get the idea into innovation, you need to follow the framework, that is, generate a creative environment to get an idea where you need to discuss your creativity with the expertise or with the layman, where the suggestions and comments has to be taken. then discuss these ideas for its usability and feasibility. So, how much it is required for the society, or how much it is required for the people in the field of medicine, and how much it is feasible to be executed. Then filter all unnecessary, so called non feasible or less usable ideas, select the best out of it, then try to achieve by providing the proof of concept. So you need to have the desired outcome based on your idea. So build a prototype version, then perform the calibration as well as validation of the prototype version. Then later on, evaluate the reliability, that is called post validation trials of your prototype device. Then you go for the permissions or the concerned regulatory authorities, then plan for dissemination. So here, this is a schematic representation of the steps in innovation. First of all, in phase one, initiate the opportunity, that is, get a creative idea. In phase two, you conceptualize yourself the prototype version or the proof of concept. Then you look for or find out the feasibility. In phase 3, you make the prototype device and calibrate for their desired outcome. In phase 4, you have to validate and verify it for their results, whether you are getting the consistent results or the accurate results, whether you are uh, doing it same time or doing by the multiple people, you should get the same result. So you need to verify for their validation as well as verification. Once everything is done, appropriate certification has to be taken from concerned authorities. Then you have to launch the product. Even after launching the product into the market, the feedback from the public, so called the medical fraternity, you have to take the post marketing trial, that is called post launch test, has to be conducted. If there is any technical glitches during the execution of the device or anything, has to be corrected in the subsequent versions. So this is the picture showing that factors influence your innovation. So here there are three types of factors can be influencing your medical device innovation. The first thing, the emotional component from the people side, that is based on the desirability of the human factors like user experience or interactivity or communication, whatever the support you people are providing to the public. Then the functional factors like how much it is feasible, the physical product or the digital product or the core intellectual properties, then the system, the technical support, what you need to provide, the whether it is user friendly device or you need to follow a structured protocol. Based on that, you can make the business where the variability. So the business model of the manufacturers, what you provide the insurance or the permissions from the government sector or the feedback from the healthcare sector. So this will make you to provide the efficient service. So if you look at this picture, probably you'll understand the desirability, feasibility, and variability. Desirability are the emotional experience of the people, the feasibility or the functional abilities of the device, and the viability, that is the marketing values. So how much revenue you can generate based on your innovation. So all these factors can influence your medical innovation in terms of execution. So this is not that easy as we think in the previous slides. We may think that, oh, okay, it is only five phases. Easily we can come across through this, the creative idea into the execution. No, it's not like that. So the innovation is not that simple because when you think of it, it looks to be very easy, but when it comes to the execution, you may fail at any point. So you should not be discouraged. Rather, you need to show your perseverance in order to work on the same idea by correcting the technical glitches or the functional issues repeatedly in order to achieve the success. So here, once again, this schematic representation will show you the innovation. Usually during the process of execution, we may be failed. When you are failed, you have to learn from the failure where you have failed. So it has to be corrected or it has to be improvised. By doing repeatedly this, at one point you will achieve the success. So in spite of taking all these preventive measures, still will be failed during the process of innovation. So here I would like to explain how to assess failure in innovation. I would like to divide into three types. The first one, qualitative assessment of failure, where the emotional and psychological components are involved, like the people may discourage you. This has already existed. Why you are working on it? It may not be work out in the execution, or this has not something novel idea. So people will try to discourage emotionally as well as psychologically. Then, coming to the quantitative assessment of failure by number of end users. Even if your idea is so novel, if that doesn't require for many people in the healthcare system, ultimately you fall into the failure of innovation in spite of having its good rationality. Then, of course, the most important, the financial assessment of failure. That is, even when you make multiple devices, even when you have a good relation in the market, if it can't generate the enough revenue, obviously your idea is no longer success. So it is considered as failure. This is the picture showing the possible causes of failures in medical innovations. That is, fear of taking risk. So when you are not ready to take a risk of your innovation, obviously your innovation is going to be failed. Or lack of market orientation. So where you need to project this instrument or where it will not work. If you don't have proper idea, obviously you will not proceed further. Then if you fail to anticipate the margin of the market, then this is not going to be succeeded. Then poor organizational structures as well as process. So whatever you have the team, if they are not collaborated or if they are not working efficiently, what you created, the idea will not be executed in reality. Then because of the wrong suggestions in when you are in anger or when somebody is suggested, so if you take a wrong decision, obviously your idea will be deviated, which will give something else which is not required. Then lack of internal communication, this is where you need to be very focused. That is, you need to have an expertise suggestions where you have to take constantly to see the updating or the stage wise improvement of the device. Of course, the most important, low priority for innovation, because there are certain devices where it will be used for the low prevalence diseases. So they are very less in population. Even if you make this idea workful, they are not enough number. So it is going to be failed. So I would like to categorize the failures in innovation into four types: technical failure, institutional failure, marketing failure, and social failure. Please do not confuse that every failure has the same reason. So here, the reason for failure is different in different innovation. Based on your requirement, it might be technical failure or institutional failure or marketing failure or social failure or even the combined failures can be seen. So let me start with the technical failure. Usually, the innovations are going to be failed because of the presence of technical glitches in the execution or the technological limitation which will be used in the supporting system or the poor testing before releasing into market. This is more important. You have to have the critical evaluation before it is going to be released into the market. So that is why the validation and verification by yourself as well as the concerned authorities has to be given utmost priority before it is releasing into the market for their reliable results. And moreover, this device may not be acceptable at this current trends. So the too much advancement in the technology of your innovation, which may not be accepted by the market, or if there is any kind of defect in the design that is called anatomical fault in the design, which may end up with the technical failure. So the institutional failure has lack of sufficient infrastructure when you don't have a proper setup of developing the innovative devices, inadequate capacity, that is, if you have more number of orders than the people who are working on it, and lock in into existing regime or trajectory, that is, people do not come out of their comfort zone or the standard instrument. So they don't want to go with the new kind of innovative ideas. So they lock in the existing trajectory or inadequate institutional framework. So this is the commonest causes for institutional failure where it will be resulting in refinement. This is where most of the people are getting suffered that where to take permissions, how much time it requires, and what are the bodies which will certify the medical devices, how much money we need to spend on it, how much energy we need to put on it. So most of the people do not have an idea on this. So for this reason, wherever you are doing the innovative conceptualization, you need to read the proper rules and regulations of the FDA, the Food and Drug Administration of America, or the Indian certifying bodies, wherever you are working, you need to have a clear idea on the regulatory bodies. Then the investors and collaborators will not have a proper agreement where the budget may cross than what you anticipated. So there is miscommunication which may result in the social failure and lack of experts advice. So you think of the innovation which could be executed so easily, but the scientific knowledge which is required for that to produce reliable results definitely requires an expertise advice. And getting approval is not easy from the certifying bodies as they require to have uh, the reliable results, the authenticity of our medical innovation, definitely it is going to be a time-taking process where usually the innovations are going to be fake. But after crossing this, even at the marketing level, if you don't work efficiently in order to make sure your device in the publicity of the healthcare system, again you are going to be failed. So the poor perception of market competitors. Don't think that you only are getting the innovative ideas. The same idea might be getting the by the other marketing competitors. So if they are very quick to produce the medical innovation to be launched into the market, of course you are going to be failed. And if your innovation is launched in a wrong time, because if that doesn't suits for the current trends, or if it is economically so expensive, which may not able to make sure your device is worthy. And if you don't give enough advertisement of your efficiency of your innovation, obviously the inadequate marketing will result in the failure of your innovation, which may not reach to the doctors or the healthcare providers in the healthcare system. And of course, the problem with most of the innovations is how much price to be quoted, because the people who are using these medical innovations are very less in number compared with the normal technological innovation. So here you need to have a clear idea how much you invested, how long you are going to work on this field. So you have to have a proper pricing idea. Otherwise, inaccurate or expensive pricing results in failure of your. So let me explain where it is going to be failed in the process of innovation by taking my own research project as an example in the innovation. So here there is something called dynamic pupillometry, which reveals the pupil response and reaction to the bright light. So it is the commonest clinical routine clinical examination in the field of medicine as well as ophthalmology and the neurosurgery department. So the application of this pupillary response in the field of ophthalmology for retinal examination, in the field of neurology to localize the lesions associated with pupil, in emergency medicine to know the brainstem activity, in the surgery department in order to identify the brain tumors, in psychiatric department to identify or to assess the impaired cognition as well as the intensity of Alzheimer's disease, as well as to know the autonomic nervous system activity in conditions like autonomic neuropathy. So conventionally, the pupil response is evaluated using swinging flashlight test, also called pen torch test, which is a subjective assessment which will provide the categorical data. So based on the magnitude and rapidity of the response, we divide into grade 0, grade 1, grade 2, grade 3 like that. So this is a pure qualitative assessment which may not be so efficient for the clinic. This is where numerous scientists have been come up with an idea to provide the videographic recording of the pupillary light response using a camera, and this can provide the duration of contraction, the time taking for contraction, the velocity of contraction. So all the variables which can be provided objectively just by recording the pupil response. However, these devices are very expensive, which will not, which did not come to the Indian market. So there is a simple scientific knowledge, that is the pupil in our eye is respond to the light, so-called visible spectrum of light ranging between 400 nanometers to 700 nanometers. So to this VIBGR light, the pupil is getting constricted called meiosis. But if you focus the light like infrared light, which is beyond the visual spectrum, the retinal cells do not respond to that. So there is no kind of pupillary constriction where the pupil itself is in a dilated state called mydriasis. So this simple idea can be used to record both extremes of the pupil, that is maximum dilated pupil as well as maximum constricted pupil. So using this scientific knowledge, one of our professor got an idea to build a dynamic pupillometry device. For that, we would like to prove our concept, that is, we had an infrared camera which was fixed to a mounted stand, and a subject was brought very close to the camera with a spatial distance of 7 centimeters. Then we switched off all the lights in that room to record the dilated pupil, because we cannot see in the dark, but the infrared camera can see in the dark. So the pupil dilated state can be captured by infrared illumination of that particular camera. Later we focus light where the pupil respond to it and leads to maximum constriction. So the both images can be captured using this camera, and that is how we could provide the proof of concept. Later, the captured images has to be measured for its diameter. So here we use a software called ImageJ software, which is an open source software for non-commercial purpose. So this software helps us to measure what is the diameter of pupil in relation to the diameter of iris. So this is the picture showing that two pupil images of two different light sources. So the one which is on the left side is maximum dilated pupil in total darkness using infrared light, and this is maximum constricted pupil in bright light. So with this initial outcome, we got into the process of execution where we would like to design a prototype version of pupillometer. So we had selected a virtual reality box to create the darkness. So we dissected the instrument in order to fix the camera. Then we have selected the two light emitting diodes. Two are infrared light emitting diodes and one is blue lighting emitting diode, and the camera where it is also dissected in order to make the infrared camera, and the capacity of this web camera is 30 frames per. So this is how we had designed this instrument, where we keep the virtual reality box with inbuilt camera to the head of the patient. So the light control can be controlled by the controlling module, and it will be directly connected to a laptop where the pupil size can be visualized in real time on the computer screen. Please remember, when you are making this device, you need to follow the certain standard procedures. That is, whatever we had selected, the infrared LEDs, which will fall in the range of 0.7 to 1.4 micrometers, which are usually present in the remotes, that will not affect our uh, eye problems. But if you cross this, then definitely there would be a danger consequences will happen. So we had gone through meticulously the procedures of the illumination, what we are trying to give in the pupillary light. So here I request you to observe the pupillary light response, which will take 3 to 4 seconds to come up. So initially, whatever you are seeing, the pupil is totally in darkness. You can also observe in the pupil there are two reflections of IR LEDs. Later, the pupil is getting constricted by flashing the white light, which will cause the constriction of pupil. So in this, how fastly the pupil is getting constricted, how much time has been taking, what is the velocity, everything can be calculated by analyzing each pupil image. So we made an image processing algorithm where initially a pupil image will be taken, and the pupil edges were identified, and that is binarized. Binarization is nothing but conversion of color print into black and white. Then the elimination of LEDs reflection was removed by filling the pupillary circle. Then we found out the pupillary outline. Later, the background noise can be removed in order to obtain the absolute pupil. This pupil diameter is easily measurable using ImageJ software, as I already mentioned, which is open source. So this is what I mentioned, what we got the creative idea to be executed in terms of desired outcome. So the entire recording of pupillary videograph can be analyzed for its diameter. So all the images diameter, if you plot as a graph, you will get this graphical plot which can be divided into multiple variables. So this is baseline pupil diameter, minimum pupil diameter, recovered pupil diameter. This is the time taken for constriction. This is the time taken for dilation. Then it can be easy for us to calculate the constriction velocity, the dilation velocity, the constriction amplitude, dilation amplitude. So here the whole graph can be divided into two phases, especially. Phase A is parasympathetic stimulation, which is causing pupillary constriction in response to light. Phase B, shifting of parasympathetic to sympathetic activity. So once you remove the light of the bright light, the pupil is redilated back to its original state. So based on the changes that occur in these three phases, you can identify the problem, whether it is by parasympathetic failure or by sympathetic. So these are the variables that are extracted from a single pupillary light reflex graph. They are latency of reflex, all static diameters of pupil, the constriction amplitude, duration of constriction and dilation, dilation amplitude, velocities of constriction as well as dilation. So all these parameters are established with their normative data and the indicators of autonomic activity, whether it is sympathetic or parasympathetic. Then we have extended this instrument into the diabetic patients in order to identify the diabetic autonomic neuropathy. So here I am projecting two graphs which were recorded from a healthy participant as well as diabetic patient. So if you carefully observe, there is so much of difference between a normal response to diabetic response. So all the changes were compromised in diabetic patients compared to the normal pupillary light. So these are the graphical plots of the same visual responses. So if you observe the blue line, which is from a healthy participant, different from the diabetic patient. So here you can see all the static diameters of the pupil as well as the constriction and dilation velocities of the pupil in diabetic patient are affected by the neuropathic changes which is deviating from the healthy participant. So this is how you can provide the reliable results in different cases in order to produce the reliability of the dynamic pupillometer. A small introduction to our own innovation, you can assess what kind of failure it is. In fact, it is not a failure, rather, we are in the process to be succeeded because the innovation will not be executed so easily into the market after processing through the certification process. So the product is not yet released into the market due to the technical improvisations which we are taking as well as the expertise suggestions, and we are also anticipate its requirement in the field of medicine. We are applying for the approval authorities for its legal acceptance. We are collaborating with the investors and creating the new devices. Explore the marketing suggestions in the appropriate healthcare system where this device is more efficient and where people will utilize it to the maximum, and keep an eye on users' feedback and improvise in the subsequent versions. So, the take home message of this discussion, in order to overcome failures of your medical innovations, find out the rationale and create idea accordingly. Take expertise advice and do not hesitate to produce the proof of concept where you need to have the definitely the scientific suggestions. Validate your device critically for its outcome. Manufacture the prototype version. Again, you need to validate for its outcome for reliability as well as accuracy. Get certified from the concerned authorities to be manufactured, disseminate it in the market and get feedback on its performance. If it is not doing well, definitely you need to change or improvise as per the need of the healthcare providers. Take care at every step in order to avoid failure of innovation. So finally, I would like to express my sincere thanks to the respected authorities for giving me this opportunity to express my view on the reason for failures in medical innovations. Thank you. Thank you so much.